Bismillahirrahmanirrahim. Assalamu alaikum students. Hope you all will be fine and safe. This is lecture number 2 and week 2. In this lecture, we will discuss the characteristics of enzymes and uses of enzymes in detail. Before starting the lecture, we just go through the recap of the previous lecture. What are enzymes? As we know, the term enzyme was first introduced by a German physiologist Wilhelm Kuhne in 1878. And according to him, enzymes are the globular proteins, and like all proteins, they are made up of long linear chains of amino acids. And whenever these chains they fold upon one another, they produce a three-dimensional globular protein. As we know that enzymes they act as biocatalysts. But there is an important thing that you must remember. All biochemical catalysts are not proteins. There are some other molecules like RNA molecules. They also catalyzes the reactions like proteins. Now, the characteristics of enzymes. The first one is proteinaceous nature. Almost all enzymes they are proteins, that is they are made up of amino acids. So all the enzymes they are proteinaceous in nature. The second one is the rate of reaction. Most of the enzyme reaction rates are millions of times faster than those of uncatalyzed reactions. It means that biochemical reactions that are controlled by enzymes they are more faster as compared to those biochemical reactions that are not catalyzed or controlled by enzymes. The third one is specific nature. So enzymes they are very specific in nature in two ways. The first one is they are specific for the type of reaction. The example is there are enzymes that are involved in the process of photosynthesis. So those enzymes they only work in the process of photosynthesis. They will not work in the biochemical reaction of respiration. So that's why they are specific for the type of reaction. Number 2 is they are specific for the nature of their substrates. Only specific enzymes they will bind with the specific substrates. For example, amylase enzyme, it will always bind with the starch molecule. It will not bind with the proteins. Similarly, lipase enzyme, it will bind with the fats. It will not bind with the proteins. Next is the active site. So what is an active site? In an enzyme's structure, there is a small portion that is involved in the process of catalysis. So that small portion or catalytic region is called active site. This active site is actually made up of amino acids. What is the function of the active site? It recognizes and binds the substrate and then carries out the reaction. As you can see in the picture, there is a substrate and there is an enzyme. You can see the active site of the enzyme. This active site, it will actually bind with the substrate and then a enzyme substrate complex is formed. After the formation of enzyme substrate complex, the reaction goes on, and when the reaction complete, the products they are released from the enzyme, and the enzyme it can be reused again for the next process. So enzymes they have specific small portion that is called catalytic region or active site. Now the next characteristic is use of inhibitors and activators. Enzyme production can be enhanced, it can be increased, or it can be stopped or diminished by a cell according to its needs. It means that whenever a cell feel needs, it starts to produce the enzyme. And whenever the cell feels that there should no need of enzyme, it will stop or diminish the enzyme production. How the cell it will enhance or diminish the enzyme production? It will do it by two ways. The enzyme activity, it can be regulated by inhibitors and activators. The word inhibit means to stop, and the activate means to activate, to start. So what are enzyme inhibitors? So enzyme inhibitors are chemical molecules that bind with an enzyme and it decrease its activity. What are enzyme activators? Enzyme activators are chemical compounds or molecules that bind with an enzyme to increase its activity. The examples of activators include hexokinase 1 and glucokinase, whereas the examples of inhibitors include drugs and ribonuclease inhibitors. The difference is present on your screen. You can easily understand from it. Now the next one is the cofactors. Some enzymes do not need any additional component to work. However, other enzymes they need non-protein molecules or ions called cofactors. So what are cofactors? Cofactors are non-protein molecules or ions that will bind with the enzyme and helps in the enzyme activity. Cofactors, now the types of cofactors. Cofactors can be inorganic or it can be organic. Examples of inorganic cofactors include the metal ions like sodium, potassium, magnesium, copper, iron. All these are the examples of inorganic cofactors. Organic cofactors includes like flavin and heme. Now the types of organic cofactors. There are two types of organic cofactors. One is the coenzymes and other one is the prosthetic group. If organic cofactors they are loosely bound with an enzyme, they are called coenzymes. What is the function of coenzymes? Coenzymes they transport the chemical groups from one enzyme to another enzyme. Some important examples are the vitamins and or compound derived from vitamins like riboflavin, thiamine and folic acids. These are the examples of coenzymes. The second one is the prosthetic group. The non-protein component when tightly bound to the apoenzyme by covalent bonds is called a prosthetic group. Now the last one is the metabolic pathways, last characteristics of enzymes. So what are metabolic pathways? To complete a metabolic reaction, different pathways are involved. And for the several enzymes, they work together in specific order in these metabolic pathways. So at every step, there are specific enzymes are involved. In a metabolic pathway, one enzyme, it takes the product of the other enzyme as a substrate. And whenever the reaction is completed, the product is passed on to the next enzyme. It can be better understood with the help of the diagram given on your screen. Here you can see the pathway number 1. In pathway number 1, there is a substrate A and there is enzyme 1. The enzyme 1, it will convert the A into product B. Now B is the product for enzyme 1, but for enzyme 2, it will act as a substrate. Now the enzyme 2, it will convert the B into product C. C is product for enzyme 2, but it acts as a substrate for enzyme 3. Now the C is converted into D and product D of enzyme 3 act as a substrate for enzyme 4, and in this way reaction is completed. Enzyme 4 convert the product D into product E. There is another pathway also. The product of enzyme 2, that is C, is converted into by another enzyme 5 into product F, and this F, it acts as a substrate for enzyme 6, and enzyme 6 will convert it into the product G, and in this way the product is passed from one enzyme to another enzyme. Now the next topic is uses of enzymes. Enzymes, they are extensively used in different industries for fast chemical reactions. As we know, they act as biocatalysts. The number one is the food industry. In food industry, what enzymes do, they will break the starch into simple sugars, and these simple sugars, they are used in the production of the white bread, buns. They are used in the enzymes are used in the production of yogurt, cheese, and other products. Next is the brewing industry, and in brewing industry, enzymes, they break the starch and proteins. Then the product of starch and protein, they are used by yeast for fermentation, and then alcohol is produced. Number third is the paper industry. Enzymes, they break the starch molecules and then they lower its density or viscosity. Whenever they lower its viscosity, it will help in making the paper. The last one is a biological detergent. Some enzymes, they are used in as a biological detergents. For example, protease enzymes, they are used for the removal of protein stains from the clothes. Amylase enzymes, they are used in dishwashing to remove the resistant starch residues present on the pots or dishes. Now this lecture is over. Hope you will understand it. If you have any query, you can email me and you can ask questions from me. Now a little bit recap of this lecture. In this lecture, today's lecture, we have discussed about the characteristics of enzymes, and we have discussed the different uses of enzymes. So this is from the page number 108 to 109 of your book, textbook. Now there is an assignment for you. Please write down all the possible questions given in your exercise in your notebook. Thank you.